15 billion years ago, a burst of matter and energy created our universe. The first planets appeared much later, and on one of them, life emerged. It developed rapidly, and the first little cells lost in the oceans gave birth to what would become flora and fauna of unlimited variety and richness. From very early on, man began to examine the mysteries of life. During the 17th century, Antony van Leeuwenhoek notably improved the microscope, and biology made a giant step forward. A century later, Carl von Linné was the first to attempt a systematic classification of all living species. The mechanisms of evolution, discovered by Charles Darwin, allowed man to understand how life evolved from simple plankton to modern man. During the same period, Gregor Mendel understood the basics of genetics, the inheritance of traits from one generation to another. A century later, the discovery of the DNA structure propelled biology into the digital era. The architectural plan required for every living organism to develop and survive is contained in a simple double helix, the code of life.
Another digital revolution occurred in the 1950s, the birth of the transistor and the development of integrated circuits, the first steps leading to computer science. The reign of huge and costly computers had begun. The coming together of computer science and biology, both in full expansion, became inevitable.
At the beginning of the 80s, a handful of life scientists had the intuition that their field would undergo a revolution. Biology would be swamped by a massive amount of data.
At the time, there was a lot of skepticism. Most researchers did not believe this was going to happen.
And our tenacity paid off.
Yes, this is true. What started with a small trickle of DNA and protein sequence quickly became a flood.
Bioinformatics was born. Amos Bayrock created PC-Gene to analyze and compare DNA sequences. In parallel, he began to develop an unknown but soon-to-be famous protein database, SwissProt. His colleague Ron Apple used computers to analyze 2D protein gels to apprehend their complex data and to compare sequences. Others, like Philip Bucher and his algorithm study, Manuel Peich with his attempts to generate 3D models of proteins, or Victor Jongeneel as one of the first biologists to foster bioinformatics, understood the extraordinary potential of computers. A new era for bioinformatics began in the early 1990s during the Internet revolution. The web portal Exposé was among the 100 first websites ever created and was specifically designed for the life sciences community. During the first year, the number of monthly hits grew from 7,000 to 100,000. But in 1996, the existence of SwissProt was threatened due to lack of financial support.
We received messages of support from all over the world.
Thousands of messages. It was just incredible, and it gave us so much energy.
The Swiss authorities realized the huge importance of bioinformatics, and a publicly funded foundation was created. 2 years later, the SIB was officially born.
We were relieved and really happy.
Yes, and we were proud. We made it.
The strength of the newly founded SIB lies in its unusual structure, inspired by Switzerland's political model. A federation of academic research groups based in major cities: Zurich, Basel, Bern, Lausanne, and Geneva. The SIB group leaders are senior academic staff members of the partner universities, whilst a number of scientists working within the groups are paid by the SIB. A real symbiosis.
Each group carries out its own research work that leads to the advancement of bioinformatics. But at the SIB, we have much wider activities. They encompass essential infrastructure and service efforts. We provide databases and software tools. We help improve healthcare, for for example, through drug design. We actively support research groups, such as in systems biology. We collaborate with industry through common projects. And by teaching bioinformatics, we prepare the next generation of scientists.
It is wonderful to have so much fun and pleasure building up a resource used by thousands of colleagues. Many of them have become friends.
SwissProt is by far the most widely known service of the SIB. Every day, thousands of scientists worldwide access it to gather information on specific proteins.
Our database is like an encyclopedia. It contains information on millions of proteins from a wide spectrum of organisms and, of course, an abundance of information on human proteins.
SwissProt is about the functions of proteins, their sequences, their sizes, their similarities with other proteins, or their changes across time, tissues and diseases. It offers the most comprehensive data available worldwide.
To keep up with a wealth of new sequence data as well as experimental results, we continuously need to update SwissProt. This task requires precision, conciseness, and a stringent respect of quality criteria. Swiss quality, what else?